Thanks, Cameron, and always a pleasure to speak with Dr. Mozaffarian. Let's get into this. I'm going to be discussing monitoring the omega-3 index to improve health outcomes. I've got a picture of the only known monument to 4 presidents in South Dakota, which is where I happen to live. I'm at the medical school in South Dakota, as well as have a small company there, and I work for a health diagnostic lab as well. But this is where I hang out, not with these guys primarily, but we'll see them again later on. What is the omega-3 index? This is the picture of the paper that Dr. von Schacke and I wrote as a proposal that omega-3 levels should be considered not just a marker of how much fish you eat, it should be actually thought of as a risk factor. And this is where we propose the omega-3 index as a risk factor for death from coronary heart disease back in 2004. And it was born of course, much of the data that Dr. Mozaffarian was showing is what prompted this idea to be born, I think, over a beer, like many great ideas, born over a beer. And this is the omega-3 index cartoon of a red cell. The amount of EPA and DHA in the red cell membrane is expressed as a % of the total fatty acids in the membrane. So it's a very simple concept here. And there's 3 omega-3s in this cartoon. Out of 64 fatty acids in this cartoon, 3 of them is 4.6%, so that would be the omega-3 index, 4.6% for that person. It's very much like a hemoglobin A1C for a diabetic. It's more of a long-term marker of exposure to omega-3, and it's very insensitive to day-to-day changes. It's a very flat and steady marker. And this also, 4.6%, is pretty typical of a Western population, as we'll see in a moment. We, in proposing this, the omega-3 index as a risk factor, Dr. von Schacke and I looked at a variety of studies trying to pick what would be a reasonable target to aim for. You have to have a number that you're shooting for if you're doing risk factors. And we came up with 8% based on several studies that had been done by that time, which was associated with the lowest risk for coronary disease. 4% was a bad place to be. As I mentioned, this is about the typical place for the United States and the EU, except for perhaps Spain, which has a high omega-3 level. Japanese are up here at 9.5% or so, 9 to 10%. They also happen to have very low heart disease rate, sudden cardiac death rates in Japan, in the United States are about 20 times higher than they are in Japan. So they have I think in the Big Jealous trial, which was 18,000 patients, there were only 35 sudden deaths over 5 years in the entire group. So it was very, very low risk. So this may also be one reason why the Japanese live longer than we do in the West, 4 years longer on average. There's a little gap here. So to the question really that I was asked to talk about, why would knowing your omega-3 index lead to improved outcomes? Why? What's the logic in that? Well, certainly knowledge of health status can always motivate change, but it should only do so, it should only cause change if we have solid evidence that change actually makes a difference. You know, it's important to actually have, before you make a change, you have to know that it really is going to help. And I think some of the evidence Dr. Mozaffarian just went through is pretty convincing that it really will help. For the omega-3s, epidemiologic data showing higher omega-3 status with better health outcomes is relatively strong, as he just indicated. Clinical trial data showing that raising the omega-3 level reduces risk is somewhat controversial. And he also mentioned that again, and I'm not going to go over those studies. Thankfully, he did. So the epidemiologic evidence, I just want to mention, actually before we get to it, I just want to put a little caveat out there that you got to think carefully about epidemiologic evidence, because you cannot draw a cause and effect. Okay. I have a different slide. I've got to come back to this later. So anyway, epidemiologic evidence in support of the use of blood omega-3 levels for predicting risk. First of all, the omega-3 index is associated with the greater protection against heart disease. About 8%, which is what I showed earlier, is a level that seems to be a target level. Secondly, the omega-3 index of over 8% was associated with reduced probability for acute coronary syndromes. This was in a case control study, one of the types of studies he just discussed. So there was a significant effect there. The index is also inversely associated with the rate of cellular aging and overall mortality in a study called the Heart and Soul Study. So this is also 8.7% was the level there. Your average intake in Japan, average level in Japan is around 9 to 10%. So these are some of the reasons, plus the under 4%, 4.4% in Framingham was associated with accelerated brain aging, and we'll have a look at that data in a moment. So that's some of the associations. I guess I've skipped the slide I was hoping to have here. Dari went over these studies. I'm not going to go over them. This is the interventional studies, positive ones, some neutral ones. There really aren't any negative omega-3 prospective trials. Negative would be they actually did harm. So the worst we can say is these studies just didn't show an effect. Because of the power issues that he was talking about, we can say that they were not able to show an effect. That doesn't mean that they actually showed there was no effect. That's a different question. I did mention that the brain function or brain volume was affected. So this is a study we published just this year looking at the omega-3 index in Framingham, where brain volume and cognitive function were assessed in 1,500 of individuals in the Framingham offspring. Average age of these people was 67 years old. At the time, they did not have clinically significant dementia or cognitive dysfunction at the time. This is a terrible table that does give you the idea that we're talking about total cerebral brain volume, and anybody who had an index below 4.4%, which was the lowest quartile, had a significantly smaller brain volume than people who had an omega-3 index above that 4.4%. Also, individuals who had index under 4.4% had poorer function on several different types of cognitive function tests. The conclusion of the study was that the MRI findings, this is the brain volume, of lower brain volume in those with an omega-3 index under 4.4% represents a change equivalent to approximately 2 years of structural brain aging. So this difference in brain size between people who had an omega-3 index above and below 4%, 4.4, is the equivalent of your brain being 2 years older. Okay. It doesn't mean, none of this means that if you start intervening with omega-3 treatment at age 67, you're going to change your brain size. Okay. This is totally a case for prevention if it's anything. Therapeutically, I'm sure we can't affect this. So can information alone change behavior? This is a good question to ask. I think it's been tested in a lot of ways. There's a very small test we did in Sioux Falls, and it really wasn't even intended to be a test. But in March of 2009, there was a health fair offered by the local hospital at the mall. And they offered the omega-3 index test, and about 300 people actually did the tests. Next year, they offered the same deal again, and 81 of those same people came back. Okay? And we were able then to look and see what happened between this time and this time for those people who came back. Now, we don't know what happened to the rest of the people, but at least these folks who retested themselves, there was an increase in the omega-3 index from 4.3% to 5.3%. Well, that's something. That's statistically significant. That's fine. But can a 1% increase really make a difference in anything? Well, if we look at some of the studies that Dr. Mozaffarian just mentioned, these are the two that he talked about and showed with the dots going down. This is the reverse, looking at the actual odds of sudden cardiac death or primary cardiac arrest as the omega-3 index goes down. So we set, and actually this was his idea to make the slide this way. It was a good idea. So we set the relative risk to 1 for the people who had the highest level of omega-3, then asked the question, what happened to their risk of populations who had lower and lower and lower omega-3 levels? And the risk goes up to about 10-fold higher at the far end, equivalent to his 90% reduction. But what happened, these are not big steps, 5 to 4.3, 5.6 to 4.7. These are 1-unit steps. So if we can extrapolate from our little experience in Sioux Falls by just telling people what their omega-3 index was and telling them it's good, bad, or nothing to worry about, people actually made a difference. They changed something to raise their omega-3 just by having the information. And I think we can expand upon that in the future. Another reason that you'd want to have a blood test to measure omega-3 levels as opposed to just asking what people eat is that there is a quite unpredictable response to supplementation. This is a study, this is data from giving EPA. This is one of the studies we did with Monsanto as they were studying their SDA product. One of the control arms in that study was a group that was given 1 gram of EPA per day, pure EPA. And what we have here, this is the omega-3, the EPA level in the red cell before and in the middle and after the placebo treatment. So this is pretty stable. These people all got placebo. But in the group that got 1 gram of EPA per day, look at the variability in the response. Some people didn't change at all, and they were all in a randomized trial. We were counting pills. We believed they were compliant. Some people didn't respond at all. The average response, of course, was up here. But look at the spread in that response. So you really can't tell what level people are going to be at just because they say they're supplementing or they say they eat fish. You really need to measure it. Another reason it's important to measure it is because genetic variation plays quite a role in this. This is data we have not, I guess it's in press now in atherosclerosis. But this is from the Framingham Offspring study. We asked the question, how much of the variability in the omega-3 index in this population of about 3,200 people, how much of the variability is due to genetic factors? And they have all the pedigree analyses, pedigree relationships worked out in the Framingham Offspring cohort. So they were able to calculate. And what I just want you to focus on here is this gray fraction up here, which is about 25% of the variability is due to genes. Intake of omega-3, whether it's supplements or known amount from foods, drives about half of the variability in the index, and about 25% is driven by genes, and the other 25% we don't know what it is. So there's really how much omega-3 you take is only going to have a 50% of the effect on variability. So another reason that you want to measure it, because just guessing is not going to do it. Here we go. This is a little bit of a rabbit trail, but I think when we measure omega-3 levels, the question is how do we express it? There are a lot of different ways to express omega-3 status. So here in, again, the 3,200 samples we have in Framingham, I plotted the omega-3 index for all these samples along the bottom axis, and I've plotted a different metric called the N3 HUFA divided by total HUFA. This is something Bill Lance, I think, has tried to make popular as another way of looking at omega-3 status. It ignores all the other fatty acids and just looks at the highly unsaturated fatty acids. As you can see, there's a very strong correlation between these two metrics. The R value, the correlation is 0.95. So essentially, they give the same information. Statistically, you'd get the same relationships. If we look at the omega-3 index versus my favorite ratio, the omega-6 to omega-3 ratio, again, 0.96 is the correlation. They're really highly correlated. And so really you could use any one you wanted to. I have some problems with this ratio that I've expressed at this meeting and published, but I think the point is we can figure out a way to express omega-3 status. And we can do it a variety of ways, but it seems like it's going to be driven primarily by the omega-3 part, certainly not by the omega-6 part. We're talking about testing. There's, as of about a year ago, these are labs in the United States that I was aware of, or North America, actually. There's a couple of Canadian labs that offer some kind of either a dried blood spot or a red cell omega-3 test. One of them, actually, we tried an experiment and sent out my blood, this is an N of 1 experiment, where we sent out my blood to several labs just for fun, just to see how the omega-3 index came back. And here's the omega-3 index from OmegaQuant, the lab that we have in Sioux Falls, where we do the test in a certain way, and we get a certain number. Laboratory G, M, B, and N brought back those values for the same blood sample. So it ranged from about 4% from when mine was 10% in our work to over 14%. So there's quite a bit of variability, and there's no standardization, certainly across the country, in the way this particular test is done. So we need standardized methods. if we're going to find this useful in the national sense. This is just for fun, again, looking at each one of these dots is a study that was done either in our lab or by our colleagues in Munich, Dr. von Schacke, who has a company a laboratory that measures the omega-3 index in the same method we use. And I also have a colleague in Korea who does the same thing at Hanyang University. So there's 3 labs in the world right now that are doing exactly the same method. And these are data from all of our studies pulled together. And you can see here's a 4% omega-3 index, as we said, United States, Western Europe, typically in that area. But then you start climbing the ladder here. The Koreans, so far in our experience, have had the highest omega-3s, from 12 and 13% sometimes on average for some of these fairly small groups. These are groups typically of 50 to 100 people. But there certainly is a relationship between fish intake and the omega-3 index, but it's not completely driven by fish intake because the Koreans don't eat as much fish as the Japanese, in my understanding. So I'm not sure what's happening here. Now, Health Diagnostic Lab, this is the lab that I have been working for now for a year. This is really the first major clinical laboratory that's offering red blood cell fatty acid testing as a risk factor. So this is sort of my dream come true, that it's actually finding its way into clinical medicine. A year ago, they adopted the method that we used at Omega Quant. And very importantly, they established a viable CPT billing structure so they're able to recover from third-party payers the cost of doing this test. And this is a science in and of itself, how you do CPT billing. But they found a way to do it that works. Beginning in June, they started testing. They've done over 200,000 samples so far. And right now, they're at about over 2,000 a day that are coming in and being run through HDL lab. And I'm sure other laboratories are going to follow because it's working and there's a demand for it. So let me give you some of the data. So what we've learned, I'll do that in a moment, I guess. This is a report. This is a laboratory report from HDL. And this is the page that talks about fatty acids. And what they have on this report is, here's the omega-3 index. This is, again, my blood, which is why it's got a name on it. Otherwise, it'd be blanked out. And there's a little green box around it. If the value came in the intermediate area, it would be a little yellow box here. If the value came in the high-risk area, under 4%, there'd be a little red box. So that's how the doctor and patient can quickly see the status. We also provide information on some of the other omega-3s individually, the total omega-6 plus arachidonate plus linoleic acid. And then very generally down here, all the monos together, all the saturates, and the total trans fatty acids, which is a value that's dropping in the United States year by year, as it turns out. At the very bottom, we just give a list of some high omega-3 fish, low omega-3 fish, and intermediate. So this is supposed to be very user-friendly for the patient as well as the doctor. So what can you do when you have a lot of data? You could have fun. So here is age and sex-specific medians for the omega-3 index as a function in 111,000 people. This is the first roughly 100,000 we looked at. Here are the ends. Actually, there are about 660 samples from people who are teenagers and about 700 samples for people in their 90s, which is the extremes. But in people in their 50s and 60s, we've got about 54,000 individuals in this area. Now, these are all people. This is not a random selection of Americans. These are all people that are going to their doctor's drawing their blood and having it evaluated for cardiovascular risk factors for some reason. Folks don't all necessarily have coronary disease or diabetes or metabolic syndrome, but they're probably at higher risk, certainly than the average population is. So that's the caveat around these kinds of data. They just are what they are. We're not trying to say this is what's average for everybody in the U.S. But interestingly, for the omega-3 index, there is a pretty strong increase in the index from the teens up to about age 70 in this group. It's a 7.2% increase every decade, from these data. And interestingly, the blue line is men and the red line is women. In the teens and 20s and early 30s, women have a higher DHA. This is driven by DHA. higher DHA level than men, which we've all hypothesized would be the case based on some fairly small data that in the reproductive years, the intense reproductive years, women are going to have a higher DHA level, presumably because they convert ALA better in that time. So this is some good evidence that it's actually true, that women do have a higher level. We've also looked at the relationship between the omega-3 index and a variety of risk factors. And this is a busy slide, I realize that. Let's just start off with this one up here. This is triglyceride levels. And what we've seen here, in each one of these graphs, the omega-3 index runs along the bottom. So people at the highest omega-3 index are over here, and the lowest omega-3 is on the left side. So how is the omega-3 level related to some of these risk factors? We're not saying it's causing any of this. This is just relationships again. But we certainly see that as the omega-3 index goes up, the blood triglyceride levels go down. And this is not necessarily because people are supplementing or eating fish. This could be just biological, that a higher omega-3 level is associated with a lower triglyceride. And this is not small. For men, it goes from 170 down to about 120. It's a reasonably wide range. Also on this graph, I have two lines, and it's true for each one of these. One says HDL. That doesn't mean high-density lipoprotein. That means HDL data. That means the people from which we're drawing this data. And then this is Framingham. So the idea here is to ask how typical are the people that we're analyzing, the blood samples we're getting, how typical are those people relative to Framingham, which we think of as a fairly normative population. The same age group, same male-female relationship, male-female proportions. I don't know what the relationships are. So in Framingham, for example, the average triglyceride is about 120, and the HDL patient population, it was about 135. So they're fairly similar. The patients that from which these data come are not that dissimilar from Framingham. LDL cholesterol levels are actually lower, about 97 compared to 105 in Framingham, presumably because these patients are more likely to be on statins. That would probably explain that. But this is very interesting. The higher the omega-3 level, the lower the LDL. Now, that's not something we've seen, certainly with randomized trials. LDL will go up in hypertriglyceridemic people, but this is just essentially ecological data. So there's something about having a higher omega-3 this also may mean that people that take their omega-3 or eat more fish are also more likely to take statins, which might cause this. HDL cholesterol, there really is no relationship between the omega-3 index and HDL cholesterol in men, but in women, it seems to go up after the median, the 4%, the average level. Very interestingly, CRP and Lp(PLA2, these are both inflammatory markers. You're all familiar with CRP. Lipoprotein-associated phospholipase A2 is a more vascular-specific risk factor, inflammatory marker. And again, we see the relationship, the higher the index, the lower level of the inflammatory markers. So it makes sense. I think the best we can say is a higher omega-3 index is associated with a lower CHD risk profile. We really can't say one's causing the other. But from data like this, it's fun to ask these kinds of questions. And I think we're getting close to the end. Yes, we are. This is an excerpt from a report or a story that was written back in 2008 by Dr. Bernadine Healy, who had been, she's a cardiologist. She's passed away now, but a cardiologist who was once the head of the American Heart Association. I think she was head of the NIH at one time as well. But she got fascinated when Lovaza actually came on the market and a fish oil actually became a medicine. And so that's what prompted her to write this story. But as part of the story, she wrote some things about the omega-3 testing. She says, this is no fish story. Raising omega-3 could be as important to public health as lowering cholesterol. Think about the comparison. Reining in our nation's cholesterol levels over the past 40 years yielded great benefit for health and longevity. The change was a grassroots effort that was driven by individuals, patients who were motivated by test results, and doctors who helped to monitor and manage them. The National Cholesterol Education Program launched Know Your Number campaign. There's been several Know Your Number campaigns over the years. But she says back in 2008, who knows the level of omega-3? Same question Lindsay's been asking. And then she ends her article with this statement: before long, your personal omega-3 index could just be the new cholesterol, the number you want to brag about at cocktail parties. Presumably she forgot to say that. And I think that time has come. She didn't live to see it, sadly. She died of brain cancer this year. But she was prescient, I think, in looking forward to saying this is kind of what's coming, and I think it's true. Well, okay. Thank you very much. I think I don't know if we're on time or not.